Hello, everyone. I'm going to tell you about some science with multi-neuron recordings. I believe Nick's already told you about some of his recordings. The data I'm going to show you was also recorded by Nick, but this is going to be different studies and with a different sort of focus, more on data analysis rather than the experimental methods. First, a question: why would you want to record lots of neurons simultaneously? You know, Neuropixels gives you this great opportunity. What does it buy you? Well, the first thing it buys you is that recording hundreds of neurons at once is hundreds of times quicker than recording one neuron at once, and that's really, you know, a pretty big deal, particularly because a lot of the neurons we're looking for, as I think you heard from Nick, are very rare. You really would have to record a very long time to find them if you didn't have high throughput methods. The other reason, though, importantly, is because they're questions you just can't answer recording one at a time. That simply would not have been possible to do with the coordination of neurons, and I'll talk about that in a moment. But first, let's think about this question of recording neurons one at a time. What is it you can do with this? And analyzing cells one at a time is old-fashioned now, but it's still, I would say, the first thing you should do when you get your data. First, because it'll help you be aware of any bugs or problems in your recording, or with the timing or whatever. But second, because you get a real intuition for what's going on by looking at single cells. So the basic workflow is that you would get your cells, make some plots to see which cells correlate with which stimulus or task parameters, summarize each cell with a number, say it's sensory tuning, and then do statistics on these numbers. But you have to be careful, and I'll explain why, that when you've got large numbers of cells, there are statistical pitfalls await. Okay, so but first, the sort of exploratory analysis you'll do to visualize the data is still the most important, and your best friend is the sorted raster plot. And there's various ways you can sort your raster plots. You can sort them in the order the trials were recorded. That's sometimes a valuable thing to do. But it's a lot of fun when you sort them by latency, by trial type, by firing rate. The vertical ordering of your trials in your raster can give you a huge amount of information. So let's look at some examples, and these are from the recordings Nick told you about, but I think plotted in a different way. So here we have an example of one neuron that happens to be in V1, and this is a sorted raster. So every black dot is a spike this cell fired, and the trial number is on the y-axis, time is on the x-axis in seconds, and here blue means the time the stimulus came on, the visual stimulus came on, and orange means the time that the mouse made the first movement, turned the wheel. And you can see here in the raster there's a response to the stimulus, and it seems like there's also a response to the turning of the wheel. If we now look at it aligned to the turning of the wheel, and we sort, so the point is you've got two times you can use in your raster. The first is time zero, what you call time zero, and the second, if you're sorting by time, is the time of the event that you're going to sort by. So here time zero is the stimulus onset, and the sorted time is the time of the movement. Here is the reverse. Time zero is the time of the movement, and the sorted time is the time of the stimulus onset. And you can see now that the first response follows the stimulus onset, and the second one follows the movement. Here we're doing the same thing, but with the beep time in yellow and the reward delivery time in purple. And there's not a huge amount going on. Maybe the cell's suppressed a bit when the reward is being consumed. All right, let's look at another example. This one is from the nucleus of the brachium of the inferior colliculus, and it's got a nice illustration of this sort of thing you can see with this. So there's all sorts of structure going on in the cell's firing, which, because this is an auditory area, perhaps there's something to do with, you know, the not completely silent environment of the rig. But in any case, one thing you see there seems to be some sort of activity coming after the visual stimulus came on. Again, it may or may not be driven by the vision. But what you see very clearly is this very strong silencing after the movement. And you can see now in this other alignment, when we align to the movement, the silencing by the movement is very clear. And it does look like there's something that's being triggered by the visual stimulus, even in the inferior colliculus here. But we see more if we trigger here, aligned to the beep time. You can see a very clear response to the beep, and you can also see a very clear response to the reward delivery. And this is presumably the click clack of the reward delivery valve that's causing this response. Here's one from nucleus PO of the thalamus. Not much going on for the stimulus and the movement, even for the beep. But whoa, what's this after the reward delivery with this amazing oscillation? It turns out that this oscillation is actually not something like the hippocampal theta rhythm, which you might have thought, coming through into the thalamus. This is actually locked to licking. Every one of these events corresponds to a lick, and the licks are so stereotyped after the reward delivery at such a steady rhythm that you get this sort of pattern even in the average. Here's another example from the hippocampus. Again, we see this apparent theta rhythm, except it's not. It's a licking rhythm here in this hippocampal neuron. So these are just some examples of things that you see in these sorted rasters. And we certainly, for example, didn't know you would have this licking rhythm in the hippocampus. But it's a good thing we did these raster plots, because then we know, and we don't go down a blind alley looking for coordination with theta and things like that. And one final example from chordate futamen. This could go on forever and ever, just seeing a nice stimulus response here and a movement response. Oh, and another one, this time from primary motor cortex. Interesting thing about this one, and I don't really know how to interpret this still, is that it comes a very consistent time after the stimulus, and it's not actually related to the movement time. It seems to be really, honestly, 150, 200 milliseconds after the stimulus onset. Interesting phenomenon. All right. Okay. So last thing to say about the raster plot, which really again is the best way to analyze your single cell data, is that there's another type of sorting. Well, there are loads of types of sorting you can do. Another one I want to talk about from the pre-neuropixels days is to sort by the mean firing rate. So this is an example from prehistoric Michigan probe recordings made in rat auditory cortex. And you can see every one of these 5 bands is the response to a tone of a different frequency. Within the bands, they're just sorted by the time, the order in which the stimuli were presented. All right, so what do we see? We see when the tone comes on, very strong onset response, and often an offset response, as you'd expect in the auditory cortex. Also, sometimes, for some tones, we get a sustained response. Also something you might expect in auditory cortex. Rarely, but this was a fairly rare example of a neuron. All right, so what's going on in this sustained response? It seems to be just pretty unstructured, but if you look now at the sustained response, you take the firing rate during this period, this time period, the sustained period, and you sort your trials rather than by the order that they were presented in, you sort them by the firing rate, you see this very strong pattern that there's a huge variability in firing rate. So some trials, you have steady firing all through these 800 milliseconds. Others, you have nothing at all. And this is not what you'd get from a Poisson process. If the firing of spikes was independent across time, you wouldn't get this sort of structure. It would be even across trials. So instead, there's a lot of variability in the total excitability of this cell from one trial to another. And it turns out that's related to the brain state, be it synchronized or desynchronized. And these different plots show the firing rate of this cell correlated with the brain state as determined by the period before the stimulus. In synchronized states, you have a lower firing rate of this particular cell. That's not always the case. In desynchronized states, you have a higher firing rate, and that's true for spontaneous activity and for tone responses. And the stronger the tone response, the larger the effect of this brain state. So it's a multiplicative effect of the brain state and the tone preference on this cell's activity. And again, you can see all of this from these sorted raster plots. So make your raster plots and sort them every which way you can think of. These graphical representations are the highest bandwidth way to get information from the hard drive of your computer into your brain. So sorted graphical analyses such as sorted raster plots are really useful. All right, so this is a really important point about population recordings that wasn't quite as clear until we had these really large-scale population recordings, but now is absolutely clear that you have to be really careful with statistics. And the statistical tests you might think to do first probably aren't going to work. So, in particular, simultaneously recorded neurons are not statistically independent, right? They are correlated with each other. You have things like these changes in brain state that we just saw that have a common investment in the firing rates of different cells that you record at the same time. Why does this matter? Because most statistical tests, the t-test, the ANOVA, most of the things you would think to use, the rank sum test, non-parametric, the bootstrap, permutation tests, nearly everything assumes that your unit of measurements are all independent. So if you measure, the firing rate change of one cell in response to the stimulus, you get a number for every cell. And now suppose you want to do a t-test to see whether cells as a whole excited by the stimulus or not. If you say that every cell is independent, then you will get a false positive result. Imagine that the cells, and it's because the cells were correlated. Imagine for a moment they were so correlated they all did the same thing, right? Then you only need one trial, and by chance your cells fired more than the average on that trial. If you treat them as independent, you get an overwhelming p-value, 10 to the minus 20 or something, that this is a significant result, but it was really just by chance. So you can't treat your simultaneously recorded cells as independent. This is absolutely critical because you get absurdly high p-values that are just wrong. Another problem is that you can't treat separate moments in time as independent because there are serial correlations in the data. This can even happen for artifactual reasons. If you've got a cell with electrode drift, its firing rate slowly changes over the course of the experiment. And now if you want to correlate that, say, well, literally with the time into the experiment or with some other variable, such as, the expectation of a reward as they learn through the course of a trial, you could have a spurious correlation due to these slow changes. And a way to sanity check your data is to not just shuffle it, because that often makes these independence assumptions, but to take, say, the cells recorded from one experiment and correlate them with the behavior recorded in another experiment. And if you find you've still got statistically significant correlations, you know there's a problem. Okay, so what's the solution? The easiest solution is to use the subject, the mouse, or the experiment as the unit of independent variability. This greatly lowers your power because you have much less of these, but it's less likely going to give you a false positive result. You can also, slightly more complex, use a nested mixed model ANOVA, which accounts for this sort of variability. So you can say there's some variability within your subject. Within each subject, there's some variability within experiments, correlated variability, and within each experiment, there's some correlated variability within the cells. Okay, so that's it for single cell analyses. Now, let's talk about the sort of things you can only answer with simultaneous recordings. So first, a way to think about the data. Suppose, and this is quite common in sensory physiology at least, you're recording a population of neurons in response to sensory stimuli, and you've got a whole lot of cells you're recording. You repeat each stimulus multiple times, and then you record as a function of time. So you've got a 4-dimensional array summarizing your spike count as a function of the time bin relative to the stimulus, the repeat number, the cell number, and the stimulus number. And this is your data, this 4-dimensional array, after you've passed it into this form. So you can think of this split in various different ways. One way is that for each stimulus, you've got a 3-dimensional array of time, repeat number, and cell. Okay, so what did people do in the prehistoric days when you could only record one cell at a time? Well, what was often done, and really they learned so much from this, for example, everything that happens now in brain-machine interfaces, or a large part of that whole field, came from one paper by Schwartz and Georgopoulos and colleagues, where they recorded one cell at a time and used it to make a, quote, virtual population, which is that you pretend you record your cells one at a time, but you imagine you had recorded them all simultaneously, and you say, what would the population activity look like in this case? We'll call that a virtual population. It's not a real population because it wasn't simultaneously recorded. If you've got a real population and you want to make it into a virtual population without having to do all those recordings, you could take a real population recording and shuffle cells independently between trials to break the correlations between them. And the point is, the difference between this sort of a virtual population and the real thing is that it loses the correlations between cells. Oh, excuse me. Hang on. Where's my window?
We still see your window, if that's so.
No, it's my window of you that disappeared.
We're not that interesting.
Okay. So how is this different to the real thing? Well, let's look again at this prehistoric recordings of tone responses in auditory cortex. So here we have the response of 4 neurons, each shown independently. So here we've got the trials arranged just by the order they were presented. Again, we've got this pure tone just for one of the frequencies, and 100 repeats of the pure tone for each of these 4 neurons. And you can see what you see in auditory cortex. There's an onset response, often. Sometimes it lasts longer, sometimes it lasts shorter. Occasionally there's a sustained response. Occasionally there's both. Occasionally there's a suppression. So what you might think is going on at the population level is something like this. And if we make a virtual population, this is what you see. The neurons are ticking away independently of each other. The tone comes on, you have an onset response. And then they go back to ticking away very close to baseline, with a few of them faster or slower if they have a sustained response. And if all you could do was record your cells one at a time, or you analyze them one at a time, this is everything. This is all you can possibly know. The data, though, actually look very different. They look like this. We've got this is exactly the same data plotted in a different way, right? So this is the data plotted by analyzing one cell at a time, rasters for one cell at a time. This is a population raster. So the rows now, rather than being trials, are neurons. And what we're seeing is on just one trial, which is what we're seeing here, is after the tone comes on, you have a burst of activity, but then you have bursts of activity at other times, and you even have bursts of activity when there's no tone being played at all. These spontaneous packets of activity. So the actual population really looks very different to this virtual population that you might imagine. So now let's see how we can summarize that idea in a more formal way. And the way you do it is with the concept of conditional independence. So remember, this is our four-dimensional array of spike counts. The real and the virtual population are the same, or are indistinguishable, if the probability that you see a particular response is the product of the probability over individual cells for a given time, stimulus, and repeat. So in other words, if you know the stimulus and you know the time and you know everything else, given those things, the cells will be independent. It doesn't mean the cells are completely independent. You might have two cells that are both driven by the same stimulus, so they'll be correlated for that reason. But beyond that, they are no more correlated than you would expect from their common drive by the stimulus. And this is called conditional independence. They're independent, conditional on the stimulus. So how is this violated? We saw it was violated because you have those population activity bursts. And to look at that in more detail, I'd like to focus on actually really mainly hypothetical data, but it's inspired by measurements in cortex. And we're just going to look at spontaneous activity, because even with no stimulus, with no stimulus, the virtual population, all the cells are independent of each other. So how is the independence violated? Well, the way to summarize it is with this statistic called the population rate. And the population rate is the simplest analysis you can do on a population other than treating the cells as independently. And all you do is, for every moment in time, you sum up the firing rates of all your cells, and at every moment in time, that gives you a sum. And then you do that for every point in time, and you get this number, which is the total number of spikes fired by any cell in the population in a given unit time. And you can see here for this hypothetical data, we've got these fluctuations in activity. And you hear about up and down states or up and down phases. It's really more complicated than that, because it's not that there's a frank bimodality here. You just have various grades of correlated fluctuation in the population, which gives you this change in the population rate. So we can learn a lot just by looking at the population rate. And this equation here is a mathematical fact, but it tells us actually some important biology. So all this says is that the variance of a sum is the sum of the variances plus the sum of the covariances. This is an easy thing to prove. I'd encourage you all to do it. But we can apply this now to our neurons and see what it means when you have these fluctuations in population rate. So this here, this sum, this is the population rate. So on the left, we have the variance in the population rate, the amount of fluctuation in the population rate. This bit is the value you would expect if the neurons are independent, because independent variances add. So this here is the difference between the virtual population and the actual population. And you can see that it's a sum of the pairwise correlations. So if all the cells are positively correlated, the real population will have a larger variance in its population rate than the virtual population. And when there's a small variance, when these are equal, that's what you call a desynchronized state, where you don't have these big correlated fluctuations. And when the variance is large, this is larger than that, so the correlations are positive. This is what you call a synchronized state. Let's look at some examples. Again, this is hypothetical data. So here, we've got all neurons independent. This is a desynchronized state. Again, it's still hypothetical data. This is the population rate histogram. It follows a Poisson distribution. And this is the correlation histogram. Some of the correlations that you measure empirically are positive, some are negative just by chance. So this has a low variance in population rate and a mean of 0 in the correlations. So now if we go back to this case where we have the common fluctuations, the population rate has a high variance and the correlations have a mean greater than 0 as you expect from that equation we saw on the previous slide. So this is a synchronized state, and this is a desynchronized state. There's other ways to get desynchronized states. So these two examples here are examples where there's a low variance in the population rate, and therefore mathematically there has to be a correlation that is close to zero on average. The mean correlation is close to zero because that's what we have mathematically. But that doesn't mean every correlation is zero. Some are significantly positive and some are significantly negative. And you can get that in different ways. So this one here is like what you get in the hippocampus in the theta state. It's a desynchronized state, and the population rate has a fairly low variance. But that's because the pyramidal cells tend to have different preferred phases of theta. So there's always someone firing. A more complicated way you can get it is that you've got different assemblies of cells firing at different moments of time. And within the assemblies, they're correlated with each other. But between the assemblies, they can be negatively correlated. So you've got positive and negative correlations mixed that together give you a low variance in population rate because the mean correlation is zero. Okay, so these different brain states, and the cortical states, I should say, are not discrete. They're a continuum themselves. And in awake rats, in this example, you have a continuum going from synchronized states, where the correlation is high, and the correlation gets progressively lower, and the population rate variance gets lower as you move into the desynchronized state. So to summarize this part, the main thing you can do with population recordings that you can't do with single cell recordings is to understand these variations in population rate. And whether you want to or not, when you have these population recordings, this is the first thing you have to look at because it's the dominant source of correlations, these change fluctuations in population rate. And if you don't look at it and realize that, it's going to lead you astray. It's always the first time you see anything, the first thing you have to ask is, could this just be explained by a change in population rate? Or is it something about the individual cells independently? So this is the most prominent explanation for population structure within an area. And I think I'll skip over this stuff. Well, just to say very quickly that that a lot of the structure can be explained by it. So for example, here we're showing triggered average of population rate triggered on different neurons. So you see,
Kenneth, you're doing quite well with time, depending on how many slides you have next.
I have 10 more slides.
How many?
10.
Well, it's 5:15, and you have 20 minutes total. So you.
So I will.
Yeah.
Okay. So so what this is showing is the firing rate in spikes per second, the spike triggered population rate. So we take 4 individual cells for these 4 different colors, and we make a triggered average of the population rate triggered on the times of their spikes. And you can see most of them have got a peak, but one of them actually got a little dip. So different cells have different relationships to the population rate. Some of them are positively correlated with population rate. Some are non-correlated. Some are even slightly negatively correlated. Of course, on average, they have to be positively correlated because that's the definition of the population rate. But that doesn't mean some of them can't be negatively correlated. And this continuum here, a word you could use for it is calling the positively ones the choristers, because they sing with the choir of the whole population, and these negatively correlated or zero correlation, the soloists, because they sing at their own time. And using just that information here, you can do a good job of predicting pairwise correlations between cells. So what we're seeing on the top is the correlation between every pair of cells in a simultaneous recording. And at the bottom is what you would predict if the only thing you knew was the population rate and individual cells coupling to it. So the first thing you have to look at is the population rate if you're trying to understand the structure in a population. Okay, so what about correlations between areas? That's a question that is still very, very open and is exactly the sort of thing that NeuroPixels probes will allow you to look at. Okay, so let's just summarize it so far. The view from a single brain area. So conditional independence doesn't hold, and the main reason it doesn't hold is because you have fluctuations in the population rate, which impose these correlations on the individual cells. And these correlations are things you would not have seen if you recorded the cells independently. The size of the fluctuations depends on the behavioral state. The more alert you are, the more you're in a desynchronized state, the more drowsy or inattentive you are, the more you're in a synchronized state where you have stronger fluctuations compared to the weaker fluctuations in the desynchronized state. And the question of what's going on between areas is an open one, but you know, there's now data. So here's an example. Whoops, I'm sorry, this second paper is out of date. It's now published in Nature Neuroscience. So this is an example of some data Nick collected with 8 NeuroPixels probes simultaneously, going through mainly the thalamus. This isn't a still photo, this is actually a movie. You can see that the mouse just isn't doing very much. And here you see some rasters with one band for every probe, color-coded according to here, and depth on the probe being the sorting dimension. So this is single trial raster rather than a single cell raster. And you can see this incredibly complicated activity going on around these different nuclei of the thalamus and elsewhere. There's a huge amount of activity going on, even when the mouse is doing nothing at all. And this is really a fascinating question. What's the structure of this activity? Because this surely has to correspond to internal cognition, because there's no visible correlate. But things get a bit easier when you do have an external correlate. So when you record the behavior, now you can start to ask how much of the population activity can be predicted from that ongoing behavior. So in this case, again, it was the same set of recordings. There's a video camera pointing at the face, as you just saw, and we took the motion energy, the first difference between successive frames, absolute value, and smooth, and then do principal component analysis. And you can see the different principal components tell you different things about facial motion. So this first one is red everywhere. So anytime there's any sort of motion, you have an increase in this principal component. The second one is particularly strong over the whiskers. And the third one is particularly strong over the snout. So these correspond to different types of movement. And then the game was to take this motion energy here, found by this method. Now, of course, you'd use deep neural networks and things, but you know, principal component analysis back in the prehistory of 2 years ago was enough, and use it to predict the activity of the neural population using a low rank approximation, using reduced rank regression, and the outcome is that so this shows some the probe locations, and this shows you know, the histology, which I think you're going to hear a lot about how to do. This shows you much better ways of doing it now. Here you see for every dot a different brain area, different parts of the cortex, the thalamus, the midbrain, etc. And the x-axis is the correlation between cells, the mean correlation between cells within that single area. And the y-axis is the mean correlation of cells in that area with other areas. And you can see that they're all below the diagonal. They're all more correlated within an area than between areas. But there are differences between them. So as an area, the soloist, the one that's least like the brain as a whole, is the thalamus. And what's so special about the thalamus? Well, if you look over here, you see the correlation with the first principal component of the face, the correlation with general motion. And you can see that nearly all of these areas have a mixture of positive and negative correlations, with the average, if anything, being negative. So when you have motion, activity tends to go down in these regions, although there's plenty of cells that also go up. So we have this structure we saw when we looked at that equation between the population variance and the correlations. You've got some positive correlations, some negative correlations, with a mean of 0. But the exception to that is the thalamus. The thalamus just goes up. So there's something different about the thalamus. When you have motion, thalamus increases in activity, almost uniformly, other brain areas go up and down. So if we now ask what's going on, it turns out you can actually predict what's going on very, very well, because from the face motion, not perfectly, not everything, but very well. If we see here, this is the activity recorded from these different regions, sorted now on the y-axis. So again, this is a single trial raster, but it's a very long time scale. That's 1 minute here. And the y-axis is the neuron number, but these neurons are sorted by a manifold embedding algorithm that puts similarly correlated neurons close together. And that's just to reveal the structure that you see in these stripes. All right, so this is the actual recording. And if we predict it from the face motion, you see it really looks very similar. It's not identical, right? But it looks very similar. It would be amazing if it was identical, because that would mean the brain is doing nothing except representing what you can see in the face. But it's still similar. And if we do I'll skip these two. Oh yeah, no. Well, here's the percentage of variance you can explain, and you can see it goes up to 10, 20, even 30% of variance can be explained by face motion in the thalamus, which again is the one that has these strong positive correlations. And if we do an analysis of the time scale, you can see that it's over an order of a second or so, less than a second, maybe 200 milliseconds when we use this fine timescale analysis, that the correlation with the face is happening at a sub-second scale. Remember we saw these up and down states in the spontaneous activity. That's not the same thing as this correlation to face motion. So if we see here, this is again a raster of this simultaneous NeuroPix data on the x-axis. On the y-axis is the cell sorted by this manifold embedding. And on the top, I'm sorry the font is so small, I hope you can see it on your screens. On the top, you see the face motion energy in these principal components. And the mouse isn't doing very much here. It's doing almost nothing. Then it starts to do something. Then it really starts to do a lot. And you have all of this structure going on. If we now zoom in to this region, you can see there's a lot of up and down states in this region where there's absolutely no motion going on at all. So the up and down states that we see here in the visual cortex are not to do with face motion. They happen even in the absence of face motion. Whereas here, when there is motion, the up and down states are actually reduced. So when there's no motion, that's when you have these synchronized states of the internally generated activity that happens on a much shorter time scale. And that's what's summarized here. If we look at the correlation of one cell with another, so this is the correlation matrix of cell versus cell, with red meaning positive correlations, and it's neurons by neurons. And if we look at it for different sized time bins, we look at 50 millisecond time bins, you see nearly all the cells are positively correlated with each other, as you would expect from up and down states. They're all going up and down together, which is why you have this variance in population rate. But now if we go out to longer time scales, you see that we're starting to get this separation into different populations which are positively and negatively correlated with each other. This here summarizes it in autocorrelations of the different principal components of the population activity on a log scale, but it's clearer if we see it here. So what we're seeing here is the prediction of one neuron from its peers, from the other simultaneously recorded neurons, and you see a very sharp peak, which if we zoom in, you can see is around 50 milliseconds in width. It's less than 100 milliseconds in width. It's a very sharp peak. But if we predict from face motion in blue, you can see a much broader peak over the course of a second or more. So the spontaneous activity is complicated. It does reflect the animal's behavior over the time course of seconds that the animal's behavior goes over. And it's a little bit stronger at a few hundred milliseconds, but there's plenty of structure at the time scale of seconds, whereas the brain is also making all of this activity at a time scale of tens of milliseconds that has nothing to do with what's going on in the outside world in the face. It is presumably things like thought. And this is just another way of showing the same thing. This time,
can just say you are a little bit less than 5 minutes away from the end.
Okay, so I will just summarize so we can have some questions. So the global picture, there are activity correlates everywhere. This is a huge caveat for your recording. So if you're looking at a behavior task, right, the fact that activity anywhere in the brain correlates with action is a huge caveat. Suppose you saw a reward signal. Well, maybe it's licking. Maybe it's the correlate of the licking. Suppose you saw a choice signal. Maybe it's a change in the non-specific movement correlates that this cell just happens to be driven by motion. This is particularly a problem if you have a go, no-go task where you're comparing movement with no movement. It's a bit better in a two alternative choice where there's always one movement or another, as I think you will have heard from Nick. Is there a correlation with cognitive state? Well, maybe, but maybe the correlation can be explained by a change in spontaneous movement. The activity correlates are not up and down states. They are slower. They happen over the course of seconds, but faster spontaneous patterns do exist, such as the up and down state. All right, so to summarize everything, the methods for analyzing population data are in their infancy. The best thing is always to start with single cell analyses, make sorted rasters, and remember that simultaneously recorded cells are not statistically independent. And also remember that motion correlates are everywhere, and have fun, because this is just such a great sort of data to have. We're going to finally figure stuff out. All right, thanks.
Well done, Kenneth. You're 2 minutes away from the end. Let's all try to see if we can make this clapping thing work. I don't know how to do it, but there's a way in Zoom to clap hands. I don't know if people can.
Maybe I'll just take it on trust that people would have wanted.
Imagine that people are happy. Next, I'd like to say just a bit of housekeeping. Thank you for observing the difference between observers. People cannot clap in webinars, apparently. Sorry. Thank you for observing the difference between observers and none, but we think we no longer need to make the difference. We were worried that there would be so many questions and so many technical issues, but from now on, everybody in the audience, feel free to ask questions in the Q&A. No more distinctions between anybody. Sorry for imposing it in the beginning. We just didn't know how it was going to go. So we have at least one question for Kenneth, which is, what is an equivalent stat to the nested ANOVA for non-normal distributed data like spikes?
It's a great question. Okay, first thing, I think you can get away with an ANOVA, right? So yes, they're not normally distributed. Nothing's ever normally distributed, really. but there aren't huge outliers, right? The time that you go badly wrong using parametric statistics is when your data has massive outliers. That'll completely ruin everything. Spikes aren't like that. If anything, they're sub-Gaussian rather than super-Gaussian. So I think ANOVAs are not too bad in terms of doing these sort of statistics. Or put it this way: if you assume your neurons are independent when they're not, you're going to get a false positive with a p-value of 10 to the minus 30. If you assume something Gaussian when it's not, you get maybe a false positive of p equals 0.03. So it's not that bad of an assumption. Having said that, there may be some sort of Kruskal-Wallis version for nested mixed models, but I'm not aware of it. You could certainly convert them to ranks before you run the analysis, but then you're almost doing exactly the same thing as a regular ANOVA anyway with the spike count. So I personally wouldn't worry too much about the non-normal.